Hey boys, welcome to the session. We're going to see our first class here, and here I'm going to tell you about the carbohydrates, its production, and its significance. Would also like to know why study about this is very important, because carbohydrates belongs to that class of compound which is also a biologically active. So here the introductory part is going to see how it is classified, and how complex it is, what is the hydrolysis product. That now I'm also using the board to explain. So let me get back to the board. So it's there. So I think now I'll be able to see the screen. So here is the carbohydrates. Carbohydrates are those class of compounds which are biologically active, and biologically important. A carbohydrate usually is hydrolyzed. Carbohydrate is hydrolyzed. So you know what is meant by hydrolyzation? Hydrolyzation is we represent it by means of simple H plus and H2O. It gives rise to CO2 plus H2O plus some amount of energy. And this is the energy that is we utilize daily. For our routine works. Now you can see that carbohydrates can be classified as complex carbohydrates, simple carbohydrates. In this you will have classified as the monosaccharides. Here you classify as disaccharides, polysaccharides, these two classified here. This is the one. A very simple example for this is the glucose. Here you will have sucrose, starch, etc. Can you see the screen? I think you are going. Right. So I assume that you are using the screen. Now within this glucose, this will take this first glucose as a very simple structure which is having C6H12O6, a molecular formula. And this is what is some hydrolysis gives you the carbon dioxide, the gas, and water, and etc. Suppose if you have too much amount of this CO2, and sometimes under suitable conditions of pH and temperature, this CO2 may react with water. CO2 plus H2 may give rise to some sort of acid. You can see that this gives rise to some acidity. Something like too much taking of the starch, which is present in potato and rice. Potato is number one source of starch, potato and rice. So this, if you take frequently, they may give rise to, if they are not properly hydrolyzed, they may give rise to a acidity in nature. That is one thing. And second thing is, if it is not hydrolyzed properly, it give rise to increased sugar content. So if there is no control over the sugar content, people with uncontrolled sugar contents are termed as the diabetic persons. The hydrolysis is done by an enzyme present in the pancreas, which we call it as the insulin. So I think you might have heard this as an insulin, heard about the insulin. So insulin is secreted by the pancreas, and then it helps in, it's a kind of enzyme. Note that enzyme is also a kind of protein, and then this hydrolyzes this particular part and you get energy. When you get energy, and this energy is the capacity to do the work. So that is the first part there. The second part is we classify them in the form of tetrose, if you have 4 carbon atoms, pentose, if you have 5 carbon atoms, hexose, if you have 6 carbon atoms, and sometimes heptose if you have 7 carbon atoms. This is another kind of classification. Pentose and hexose are very common, aldopentose. Suppose if you have an aldehyde group there in the carbohydrates, you call it as aldopentose. Suppose if you have keto group there, you call it as the ketopentose, aldopentose. Similarly, if you have hexose, you call it as the aldohexose and ketohexose. You would want to know that aldohexose is also called as the reducing sugar, and it is non-reducing sugar. So aldohexose is a reducing sugar, ketohexose is a non-reducing sugar. It will have a keto group. This will also have a keto group, but with an alpha hydrogen. Therefore, this results in quick submission of hydrogen, then reducing the other thing. Therefore, that is why you call it reducing sugar. Okay. So here is the part that I would like to stress upon. The reducing sugars are very important in your lab. So you are going to test whether the sugar is reducing sugar or non-reducing sugar. And you are going to report if you have the reducing sugar, you are going to get several organic analysis. There is organic qualitative analysis. Therefore, what is the functionality provided by aldohexose is not provided by the ketohexose. That is to say that, just a minute, I'll just check if boys are joining there or any comment is there. Okay, boys are joining there, so that's very good. So because we are beginners here, we are beginning the class here, it may stress that it may take some time to get used to these classes. But anyway, you can raise your voice and ask questions. You are always welcome to ask them through any kind of model which you cannot ask. So this is the second part of the classification. And the third part is I told you that it is sugars having an aldehyde acetal equivalent and sugars having a ketone function or an acetal equivalent. And then the last part of the classification is the reactivity part that also is there. That is the reactivity part. Reactivity part is also obtained from here. In the reactivity part, you will have whether the sugar has the reducing capacity or non-reducing property. why do you require this? As an example, you can see that once this sugar is inside the human body, there are a lot of biochemical reactions. And these biochemical reactions are exclusively dependent on the pH. Suppose if you have a reaction which occurs at 7.2 blood, the pH of blood, which is alkaline, then, like for example, during the night, the pH may slightly increase, like 7.3 or 7.4. When the pH increases slightly, there is a production of a protein from an enzyme called as serotonin. Okay. Serotonin is a sleep-inducing chemical which starts as soon as the sun sets. Okay. Now, why I am here is the pH is initiating this particular reaction, right? So how do you initiate the pH? You initiate the pH by so many complex biochemical reactions, and one such biochemical reaction is provided by the reducing capacity of the sugars. So if you have a reducing sugar, it's well and good. If you don't have a non-reducing sugar, how do we test it in the lab? We use tolerance reagent. Tolerance reagent test is a very important test. So if the AgNO3 solution that we are using, it's an ammoniacal silver nitrate solution. You add this to carbohydrate, add some amount of NaOH, and then when you heat it over a water bath, we'll get a silver mirror. This is the confirmatory test for the carbohydrates. Note that all carbohydrates will not answer you the silver mirror test, because condition is you should have the reducing capacity, and then only you will get the silver mirror. Most of our carbohydrates which we see in our everyday life are complex carbohydrates. Okay, and it's very difficult for them to reduce initially. But once they breakdown means converting complex molecules into simple molecules. Once they break down, you get a lot of simple sugars. Okay, so I think we have classified the carbohydrates in 4 types. First thing, the complexity. In complexity, we have used the monosaccharides, disaccharides, something like that. Okay. And then the second, we have seen how it is classified according to size. In the size, we use C4, C5, C6, pentose, etc. like this. In the third, we have seen reducing or non-reducing sugar, and in the fourth one, probably that should be the third one, and the fourth one, we have to mention whether it is a keto group or whether it is a aldehyde group, compounds. Okay, so this is the basic classification of the compounds, and the example that I gave is monosaccharides is a single molecule of the sugar, which is nothing but glucose, fructose, mannose, something like that. And in the disaccharides, you've seen the disaccharides, oligosaccharides, polysaccharides. You see a lot of things in this. So I can go to a few examples also. That if we go from monosaccharides to higher saccharides, we are going to get rice, potato, and then some more complexity is there in that. You can see that will give rise to paper. Some more complexity that can give rise to cardboard. Some more complexity to that we can get hardwood. Of course, hardwood, any wood is a carbohydrate, but it has got a very good long life. The reason is they've got some oil, sandal oil, rose oil, something like that. The oil is present in between the layers of the carbohydrates. Therefore, it has a very good strength, tensile strength, and durability. So carbohydrate is also, you can see that some animals eat paper. Okay, this is using there are enzymes in those animals which convert the paper into sugars and then hydrolyze to water plus energy. Okay, carbohydrate is also such thing, but it is some multiple layers. So as you go from increase the molecular weight, which is gone increase the molecular weight, the complexity is increases. You can see what is very simple potato in the form of biological thing to the non-living thing is it's all complex with the carbohydrates. And the human beings, cannot survive without the carbohydrate. The important thing about the carbohydrate is it is a very good source of energy, and about 80% of the energy for the brain comes from these carbohydrates. Suppose you can see that biologically, if a person feels giddiness, okay, giddiness means maikammaramari. So if a person feels giddiness, it is because of his constant working with an examination or some kind of stuff or some other thing. The brain loses an amount of the sugar, and therefore it requires them in the form of some from the output. So usually what you do is you give glucon D or glucose supplied in the fruit, like particularly oranges is a very good thing because it is acidic in nature. Acidic in nature also is able to quickly metabolize that. Metabolization is quick transfer of energy from carbohydrates to the human body. That is the metabolization. So you can see that carbohydrates are also very essential in their absorption in the body. Absorption in the body. So you have several foods. Okay, I can quote a few. Rice, cooked rice, we call it as soro. It's different from the idiyappam. It's different from rice roti, and it is different from puri. And that one is puffed rice. We call it as puffed rice, and that one is different from some more form we have that we call it as the momo. Okay, so in all this we have different structure. We are going to see the structure in our next class. In all this we are going to see the structure. The structure changes, the outermost physical appearance or the 3D appearance also changes. And likewise, digestion also changes. Suppose if you have boiled rice, pacharasi or what you say, that kind of absorption is different from the other kind of the rice. Suppose if you have, we call it as the ganji, okay, it has got a very good absorption in the body because all the higher molecular weight carbohydrates, higher level carbohydrates are broken down to smaller chains and can quickly be absorbed by the body. So whenever a patient is in quick need of the energy and quick absorption in the body, it is given by this half digested rice. So I think I just want to see how many students are there in the meeting. Next time we will simply finalize. The participants, Prasanji is there. Okay, anyway, the recording will be available in the YouTube. And because this is a beginning session, I'm going to end here. And thank you for watching.